Hello and welcome to this next chapter in biopsychology. In this particular chapter, we're going to be looking at all the other senses. Not the best approach to this, I think. I think there could have benefited from a hearing chapter and a touch chapter, and at least get some of those basics there. But the textbook decided to put it all into the same one, and so we'll take a look at the rest of the senses. We spent a lot of time on vision. Let's spend a little bit of time with some of the other senses and being selective. Can't cover them all. But here we go with Chapter 7, the other sensory systems. And we begin, as we began with vision, by talking about sensory systems in general. Essentially, we're looking at outside energies that are translated into electrical impulses by specialized sensory neurons, the sense nerves that we have based on different areas of the body and different kinds of energies that we have. Now, when we think about the senses, we often think about the 5 big senses, and we've often been taught we have 5 senses. We have taste, touch, sight, smell, and hearing. And certainly we can learn what we need to learn about the basic biology by studying each one of these. There are, however, lots more senses that we have. Now, just right off the bat, if we're going to look back, we look at the energies and the actual neurons themselves translating that information into electrical symbols. In taste, we have the taste buds. In touch, we have the sensations of pressure, temperature, pain. In sight, vision, we've discussed that. The sense of smell, we have olfactory cells within our nose that identify different smells. And of course, in hearing, we have specific sensation cells within the cochlea of our most inner parts of our ear. We'll be spending some time looking at that one. Now, we have specialized neurons for each one of those, and we have specialized neurons for all of these other senses. Look at this graphic here. This is a graphic that represents our current knowledge, the accepted perspective, and it says radical. These are theories that are out there that are being explored right now in the research, that our nervous system may be a lot more complex than we know it to be right now. So essentially, however, there's evidence out there of tons and tons of different kinds of senses, way beyond, inclusive of, but way beyond the first 5. So as this scrolls, we can talk about each one of them. We have vision, sensitivity to light, also to color, but then maybe specialized neurons for red, green, and blue. Hearing and the specifics for that, different tones, different instruments. Look at this one, smell, that's fine, but we may have up to 2,000 different receptor types. Same when we talk about taste, the sweet, salt, sour, bitter, and umami is a Japanese word for deliciousness, or the sense of deliciousness. Now, touch, light, touch, pressure, those things. Pain, different kinds of pain. Cutaneous, somatic, visceral pain, when you're just near something that seems painful. And then mechanoreception, balance, how we rotate, linear acceleration, proprioception, knowing where our body is in space, kinesthesis, muscle stretch. Look at temperature, heat and cold, separate neurons. And then interoceptors, a sensitivity to our own internal blood pressure, arterial blood pressure, central venous blood pressure, head blood temperature, body oxygen content, cerebral spinal fluid acidity, osmotic pressure. This might relate to thirst, arterial vein blood glucose difference related to hunger. Lung inflation, we certainly feel that when our bladder is full. We feel that when we have a full stomach. All of these things. Now let's add some more. We also get an itch. That's a whole other sense. We have thirst, as indicated here with plasma osmotic pressure, and hunger. artery, vein, blood glucose differences that we may have sensitivity to. We sense magnetic fields. We sense electrical fields. We sense time passing or slowing down or speeding up. And of course, the various kinds of things that go along with stress. And we'll see when we really look at all, let's look at it this way. This is the input from the outside world and from our bodies that we process in order to create a very important component of our state of awareness. And that is, what are we feeling in terms of our sensations, not in terms of our emotion expression, but what are we feeling in terms of all over our body? Way more than 5. Way more than 5. So we're going to take a look at a couple of these, and let's start off. One of my favorites, and it's one of my favorites to talk about because I am hard of hearing. I am wearing hearing aids. They're specific, programmed to the very narrow band of hearing loss that I have. And so let's take a look at how the hearing process works when it's normal. Then we'll talk about when it's not normal. So here we have hearing. And just like the eye, we have a organ, our ear, the ear canal, the middle ear, and the inner ear, specifically designed for this kind of activity. So here we have a sound wave, and the sound wave travels, is funneled by the pinna, the outside surface of the ear, down the ear canal to vibrate the tympanic membrane or the eardrum. That results in the vibration of these other inner ear bones, and they vibrate at the stirrup or staples. That stirrup is connected to what's called the oval window, and it translates that vibration to the fluid that is contained within the cochlea. That starts to create an electrical signal. That electrical signal, depending on where it's being relayed, will translate down the cochlea and then out the optic nerve to be interpreted by the brain. Now, within that cochlea is a structure called the basilar membrane. Now, this membrane exists within the cochlea. It's in the same shape of that. And you'll notice that at one end, the actual structure is stiffer, and then it's sort of floppy at the other end. You can see where those labels are. Now, what this indicates is these different areas respond to different kinds of vibrations. So very high frequency sounds will vibrate toward the stiff end, the beginning of the cochlea. Lower sounds will translate further down the cochlea to the more floppy ended. And you can see the different hertz, the different frequencies. These are, 7,000 hertz is 7,000 vibrations per second, 5,000 vibrations per second, 4,000 vibrations per second, all the way down to 600 hertz, even 200 hertz as it goes down. And these are the different frequencies in terms of the pitch. In terms of really low notes have low frequencies, and really high pitch notes have high frequencies. Now, I included in the course a link to the video, Dr. K's Psychobabble video on the auditory cortex and the basilar membrane, where I go into some detail about this particular topic. But I'm going to spend a little time here taking a look at a particular graphic. Let's see if I can get this up and running here. Right here. To talk about frequency and how we hear things. Now, this audiogram is of what we would call familiar sounds and where they exist along two dimensions. Going across the top is the frequency pitch. meaning the cycles per second is hertz. So you have 125, these are really low notes, and then you have 8,000 cycles per second, which is very, very high pitched notes. And you can see in this area here that rustling leaves and birds chirping and whispering, look at that, way up high versus other parts of language. I talked about that in just a second. Water dripping, very low. That bloop sound is very low. In this sort of banana-shaped area is what's considered language. This is the primary language area. And you see the different consonants and sounds, the NG sound, the E sound, the R and the A, the ch sounds, and H and G and K's, F, S, and TH. Now, I mentioned that I have hearing loss, and my hearing loss is in this upper register here. Now, it doesn't keep me from hearing many things that are loud enough in this area, but it interferes with my understanding of a lot of components of language. So I have hearing loss, but I'm a musician. I can hear musical notes for the most part. Language, consonant sounds like the ones in that diagram are the ones that are missing from my hearing. Missing to the point that really there's no amplification that can replace them. The way my hearing aids work, and hope this is an interesting story. I was in need of hearing aids for almost my entire life, but the technology was not ready to do what these hearing aids do now. These hearing aids, in order to help me hear language better, actually sample those sounds very quickly. They take a recording of those consonant sounds and they re-pitch them at a different tone. So the CH sound or the K sound that I don't really hear very well is then played back in my hearing aids immediately at a lower frequency that I can hear, and that's amplified. Now my brain, when I first put these on, I could tell something was going on. There was something off in terms of the way language sounded, but part of the process of getting hearing aids is that your brain begins to adapt. And it begins to say, okay, that CH sound, which is supposed to be over here, is now over here, but I'm going to blend it in with the rest of the sounds, and in terms of my cognitive experience of that sound, it's going to be the same as it was before. So as my brain adapted to my new hearing experience of those particular sounds, that my experience normalized. However, if I take them off, I am way more deaf than I was before. My brain would have to readapt and become overly sensitive and focusing on those areas. Right now, it's sort of laid back. The hearing aids are doing part of the work. If I took them off or lost them and could no longer wear hearing aids, I would have to readapt to the point that I was, which was problematic. I mean, it's a disability, so I didn't have the ability to actually hear some of those words. And understanding language has always been an issue. So as we look again further on this, we see the left to right is the frequency, the pitch, but then we have amplitude, which is the up and down. So we see whispering is up here, that's very soft. Vacuuming is way over here. This is getting louder. Piano, the dog barking, it's low, but it's louder. And then down here we have like really, really loud sounds like trucks, lawnmowers, and jackhammers really down here. A rock band right in the middle, all the different sounds, the chainsaw, and when these speed bikes here, an airline, the siren, all of these things, extremely loud. Hearing loss in these areas would be severe and profound. And it happens to be that my hearing loss in this particular frequency is actually profound. I have extremely low hearing in that particular frequency. So when an airplane goes by, I don't think I hear all the high frequencies, and it doesn't bother me as much. In fact, I know that when we on stage, when we've had feedback, sometimes you get that oscillating feedback from an instrument, very, very high-pitched sound. I sometimes can't hear it. Sometimes it's so loud, I can feel it. I can feel it, the pressure on my eardrum, but there's no cognitive experience of that note because that part of the basilar membrane is not responding to that particular frequency. So that can be an interesting experience for me to have that. So here we have, I go into this a little bit in that video, and we just talked about the audiogram of familiar sounds. Let's talk about the auditory pathway, how information comes into the ear and moves to the brain for processing, similar to what we did in vision. So here we have our sound, and the sound comes into the ear and enters the cochlea, and wherever on the basilar membrane it vibrates, it sends that signal, and it sends that signal to the brain stem. And you'll see these steps as the signal is moving up. Hearing is first processed in the brain stem, not in the brain. And so we have first order processing coming from the cochlear nucleus at the right at the same level of our ears. The signal then moves up to the superior olive, which is essentially the ascending path of the auditory signal, moving over to the inferior colliculus, which is a switchboard, which is sort of, now we're starting to get into some processing, attentional issues in terms of then, parsing out the different sounds by pitch and frequency. And then finally into the medial geniculate, which directs the auditory information to the brain and activates our attention systems to those sounds. Now we can begin processing. So let's further look at this particular pathway by looking at a different graphic here. And here we have a picture of the auditory cortex is sort of on our temple, if you look at it from the outside, and the upper surface of here divided between the primary processing and secondary processing cortexes. So our signal appears in the primary auditory cortex, and depending on which note, which frequency it is, that can activate neurons all along this particular area, and then it's passed on to the secondary auditory cortex for processing and identifying the source of that information. Now notice this is similar to the visual pathway in terms of identifying what something is. Now we're identifying what something is, what something sounds. You have the visual pathway into this general area about what something is, and then now we have the auditory pathway into the same area. Now, taking a look at this particular graphic, and we can compare it to the basilar membrane, and we know that low notes on one end, high notes on one end correspond to the same low notes on one end and then high notes on the other end. There's a direct connection between the notes on the basilar membrane as they're mapped out, the different nerves along the basilar membrane, and the actual direction in which the order at which notes are translated into the primary auditory cortex. Now, right at the beginning of the course book, I introduced the notion of music as probably one of the most, for me at least, one of the most profound notions of hearing, and along with language, so music and language. And it's interesting to recognize, first off, as you'll read in the textbook, that the brain has been created in a way that it distinguishes language from music very, very early. In fact, it may be hardwired in, the difference between music and the difference between spoken word. In fact, if we listen to music from another culture, using notes and scales and intervals that we're not familiar with, we may not enjoy it because it doesn't align with our Western thinking of how music should be in harmony. But there is no doubt we will recognize it's music, and we won't misinterpret it as natural sounds or as vocalizations or language. This is built in. We can hear a language that we don't understand, but we know it's language. This is one of the ways in which we when we listen to when biologists listen to whale song, or the sounds that whales and dolphins and other cetaceans create, there's a notion, there's an inkling that we're listening to language, and it's one of the more compelling aspects of that particular field of study, that there seems to be a complexity, there seems to be an order. It's triggering all of our sensations about this is language, and of course, that is a current pursuit in trying to understand whether or not this is language and what's it saying. What are they talking about with each other? Now back to music. When we experience music, I included a graphic in the course book explaining how the entire brain reacts to music and what does that look like. And so here we have, just go from the top right all the way around, the sensory cortex is involved in the tactile feedback connected to playing an instrument or dancing. The movement that we have, our sensory cortex. The auditory cortex, perception and analysis of processing of chords, notes, melody. The hippocampus, musical memories, experiences in the context of music itself. Visual cortex, reading music and observing the source of music, of looking at different instruments and looking at different people as they're arranged on a stage. The cerebellum, connected to emotions and movement in terms of emotions, in terms of music. The amygdala also involves in our emotional reaction to music. The nucleus accumbens, involved in our emotional reactions to music. Prefrontal cortex, this is where we process this information at the highest level. Expectations are created. This is also linked to fulfillment in that particular musical experience. The motor cortex, involved in moving around, similar to the sensory cortex. And then, of course, the corpus callosum, connecting the right and left brain aspects of this. Now, I want to talk a little bit about the prefrontal cortex, about this satisfaction. I just want to highlight a particular experience that I know that you've had. Now, usually in Western music, there are 8 notes in a scale, and the root note, which defines the scale, say like an A flat major scale, the A chord, which is made up of the first, third, fifth, and sometimes the 7th chord, the 7th note, those 4 notes, or the triad of the first, third, and fifth, make up a chord. Now, when we look at the first, third, and fifth notes of the second note in the scale, the third note, they create the different minor, major, and diminished chords within a major scale. In Western music, we often talk about the resolution of the song being the time in the song when the chordal structure returns and ends on the root chord. So in A flat major, you may be playing a C minor for a while, but then when it comes to the end of the song, you go from that C minor down to the A flat major. And like dun 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 dun, and that kind of resolution completes the song, and we get this sort of visceral experience of the end of the song. Now, one of the things that as we experience more and more music, is music has a very, very cool quality of enticing emotional reactions through a process of tension and release that is often entailed in the use of music to when do we resolve, when do we not resolve, how do we put discordant sounds in there in order to create discordant feelings in the individual, and the art of creating music backgrounds for movies is presented right in front of us. In jazz music, jazz, which is considered to be a very highly energetic and conflictual type of music, is often unpleasant to people because the chords don't always resolve. Sometimes you end the song on a minor chord, and the whole audience is sort of like da-da-da-da-da-da, and they're waiting for the dum, but it doesn't come. And that kind of tension, experienced listeners and individuals who, attracted to that, like that tension. They like that the song didn't resolve, it didn't create that finality of it. And we experience musical movements in those ways. There's certain keys, certain minor, certain modes in which we look at the intervals between notes and whatnot that elicit certain emotions. So a major scale, for example, will elicit sort of a happy sort of sense. It has a happy resolution. The chords work in a way that's very happy in terms of our experience of that music. However, a lot of, let's say, sad songs are written in minor keys, so that the relation between the root note, let's say the A flat, and the third is a minor third. So rather than it being a full step to the third, it's 1.5 steps to the third. And it creates a melancholy sort of feeling to that chord. Now, again, in that video that I did on Audition, I play some of that music on my guitar. We look at chords and intervals and whatnot to get an understanding of that. But there's a real deep science to understanding the relationship between music and the brain in a whole field called musicology, which is the psychology of music. Now, I included also in your resources a link to a very, very brief article, but it links to other resources, which is a really good way to explore this, why your brain likes music. So I really encourage you to go look at that. Now, we don't want to leave the ear too soon, because the ear also contains some interesting apparatus having to do with another aspect of our sense system, and that is equilibration, or balance. And our vestibular system, which is built into, if you look at the where this graphic is, this enlarged piece is coming from the beginning of these semicircular canals that are full of fluid and nerves, here represented by these hair cells that respond to our orientation in space. So right now, gravity is pulling me down this way. If I turn my head this way, the fluid in my semicircular canals is moving. It's moving against those hairs, and information is being translated to my brain saying I am no longer horizontal, I'm no longer vertical, I'm moving horizontal, and I can experience myself moving in that way. Now, we saw this connection between the vestibular system and our eyes when we talked about vestibular eye movement, which I can move my head and I can go like this, as I did in the last chapter, and information about where my head is in space being translated through the semicircular canals in the vestibular system translate to motor movement in my eyes to help me keep my eyes focused on the object of interest. Now, this system right here, complex system built in these semicircular canals, exist within XYZ coordinates. So we have a three-dimensional layout, very similar to a gyroscope that we use for navigation. In fact, that's where we got the idea for a gyroscope. Now, some interesting factoids about this particular system that I find really fascinating. For one, within this fluid are crystals of calcium carbonate, and these are the ones that rub up against, sounded like it's a thick viscous fluid that's full of these little crystals, and these little crystals are what rub up against the hairs within this sensory system, those are made of the same building blocks of marine bones and seashells. So a very interesting aspect of how we are connected to the ocean. Now, in addition, the semicircular canals inspired the development of things like gyroscopes, which are devices used to help orient machines, airplanes, whatever, make them orient into space and time so that navigation systems know where they are. Now, the next group of senses that we're going to look at are the mechanical senses. And these are made up of three different kinds of receptors: mechanoreceptors, thermoceptors, and nociceptors. Now, in mechanoreceptors, what we have: touch, vibration, pressure, stretch. These are the kinds of sensations that we have that are specifically from those kinds of sense neurons. In thermoceptors, we might be able to guess here's where we have our experiences of hot and cold. But an interesting thing about thermoreceptors and how they work is cold receptors and hot receptors are continually firing, maintaining our experience of what is the base temperature. When the temperature goes up or when the temperature goes down, some of those will stop firing. When it's too hot, the cold ones stop firing, and when it's too cold, the hot ones stop firing. And what we'll experience then interpret that as an increase or decrease in temperature. So it's actually the absence of one of them that makes it makes our sensation. This is why sometimes we have this sort of, and it's in the graphic here, this notion of I might touch something hot, but my first experience of that is that it's cold. And the same thing might happen when I touch something really cold. It might actually create the opposite immediate experience of being hot. So we have thermoreceptors that are basically responding to changes in the environment, and that's when we get the experience. We have bursts that this can happen that allow us to pull our hand away from very cold and very hot surfaces. We have warm receptors specifically for relaying information about warm. And what I was just talking about was this paradox of, because of the way thermoreceptors work, we sometimes can make the mistake between something that's very hot and very cold. And finally, nociceptors have to do with pain. Specifically. So we have pain receptors if things become too hot or too cold. We have pain receptors if there's too much pressure. And you'll see how these relate to mechanoreceptors and thermoceptors. When those are pushed to the brink, we have other neurons that kick in relating that too much of a high temperature, too much of a low temperature, too much pressure. Chemicals that go to our body that activate these, chemical burns and whatnot. These activate when there is a near and present danger of tissue damage. This can be done by the actual temperature and pressure, but also through inflammation. And in fact, inflammation, the gathering of fluids in a particular area and the swelling of that particular area, is kind of like the go-to concept in pain management. When we take aspirin or Tylenol, they operate in very different ways, but we are, in terms of aspirin, we're actually reducing the blood flow to particular areas, or allowing for more blood flow. It's a vasodilator. Thinning of the blood as well, so there's less chance. It reduces the swelling in a particular area. Now, when we take Tylenol, it impacts the central nervous system and the processing in nociceptors in a slightly different way, but all of these as a result of inflammation. Now, the next area we're going to look at is chemical senses. So we have hearing. we have the mechanical senses, and now we're going into the chemical senses, and these are taste and smell. Taste and smell work in coordination with each other, but I'm going to spend a little bit more time talking about taste. Okay? So we're going to kind of look at the pathway that occurs when we are tasting something, and this works in coordination with our olfactory lobes, with our olfactory cells that also send corresponding information. When we taste and smell something, they activate the same areas. So I want to have our little stimulation here. If you see that yellow dot, from the taste buds on the tongue, that information translates over to the brainstem. Okay? Then it moves up the brainstem into the pons, actually, is the area of the brainstem here, and that allows for an activation of the amygdala, which is an emotional sense of this particular sense of taste, and that information is immediately translated to the prefrontal cortex for processing. Our original signal continues along and actually gets into the ventroposteromedial thalamus and then goes on to the somatosensory cortex for identification. What am I tasting? At the same time, we've also had activation of the primary taste cortex, the insula, and that sends information to the prefrontal cortex in order to identify, enjoy, spit out, whatever it is that we are eating. So that's the pathway of information across the brain when we taste something. Now, I want to refer to another video that I created. This is, again, a Dr. K's Psychobabble video on satiation. I put the link to this in the course. And this has to do with the mechanisms of hunger. And then I look at Thanksgiving itself and what happens at Thanksgiving in terms of hunger and the justification for dessert. A little theory I have as to why we have desserts. That brings us to the end of this particular chapter, and our assessments consist of, first, in the discussion, I want you to find music that you do not like, and I want you to listen to it and talk about the experiences, the emotional experiences, and this is kind of like old psychology, sort of like the, back in the day, psychologists should expose themselves to stimulus and they would think deeply about it, just self-introspection in a way of figuring out how the brain works. I want you to engage in a little bit of that. So you put on the headphones, you listen to some music you don't like, and think about the images and the context or whatever that comes up, and talk about how this activated certain aspects of your brain and what might contribute to the reasons why you don't like that kind of music. Now, in the second aspect of today's work is, the work to do at Chapter 7, is a quiz on the auditory pathway. And so describing how you would experience a sound using the information that I just outlined in the entire audio pathway, going from the outside the ear all the way up to the primary and secondary processing cortex for audio information. So that brings us to an end of this chapter, and I hope this has sort of given you sort of an idea of how really cool and interesting our sensation systems, and again, all those senses bring in. We could take those pathways and examine those 33 different senses that we have, along with the others that I put in, like itch and hunger and all that stuff, and all the rich research that's out there about the information that is coming in from our body that allow us to navigate this world. So I hope that was enjoyable, and have a great, great day.